we're going to bring the entire complex over to the ER, which is what I just went over. I went over the nitty-gritty of the actual molecular interactions that occur in that situation. Here we're going to take a big picture approach. So if this protein needs to either be inserted into a membrane, if it needs to be excreted, if it's going to end up in a lysosome, then it needs to go through the ER. Also, if it has a job in the ER or if it has a job in the Golgi or in the membrane of either of those two organelles, then it has to go through the ER. In addition, if it's going to have a job in the nucleus, then it needs to go through the ER as well, where you're going to have the modification of the protein. So what happens in the ER? We have folding of the protein. We also have addition of sugar groups and modification of sugar groups. So the folding was important. That's what we talked about here. There are things called chaperone proteins that help other proteins to fold. So we talked about a specific chaperone protein that's basically like a barrel. So this protein is called GROL, I think. So it's essentially literally just a hollow cylinder, and then other proteins could go inside here. And then there's also a cap that goes on the top of this protein. And so we have the N-terminus and the C-terminus. And when this nascent protein or unfolded protein is inside of this chaperone, then it's protected from any water molecules, any ions, or any other proteins that might interact with it and cause it to fold incorrectly. So scientists think that this protein or this peptide sequence could now change a bunch of different, or try a bunch of different conformations and to find the right one that it's supposed to be in. It's not fully understood how this happens, but we do know that it's isolated from other influences like positively charged ions or negatively charged ions or other proteins. So this is again happening in the ER. If there are a bunch of proteins that don't fold correctly, the chaperone proteins, again, this barrel-shaped protein, could send a signal to other proteins in the ER that live in the ER. They have their functions in the ER membrane, and they will sense that there are lots of unfolded proteins. This could cause the UPR, or unfolded protein response.